We have now Madan Rao, who's coming from NCBS, the National Center for Biological Sciences in Bangalore. Thank you, Madan, for coming from India. And we look forward to your talk on information in development. Thank you.
Thank you. Yeah, has to come way down. Okay. Thanks a lot, Thomas, for the invitation to speak in this symposium. It's a real honor, and I'm really excited by the talks that are presented and the discussions that follow. In my talk today, I'll discuss our ideas on information processing, computation, and control in living cells. It's something that we've just gotten engaged with. And the idea is to try to regard living cells and tissues as a distributed system. So my talk is going to intersect with the talks of Yaron and Lisa, but it's very, very significantly overlapping with the previous talk. So I'm going to skip some slides. Yeah, okay. So what I'll do is I'll discuss two distributed computing problems in cells and tissues using the following examples. First, I'll discuss how cells in a tissue precisely infer their position, something that Thomas talked about, and by decoding information from a very noisy morphogen signal during development. The motivating example is going to be experiments done on the wing imaginal disk rather than the fly embryo. Now, this will involve optimally deploying surface receptors, as shown there, and designing sort of cellular chemical reaction networks with feedback control that leads eventually to transcription. So the idea is that morphogen output, a noisy morphogen output, gets converted to a less noisy, noisy morphogen input gets converted to a less noisy output. And the second one, I'll address a related problem of precision in encoding complex information. And this will be the sort of synthesis of very specific proteins that are decorated with sugar groups that gets then displayed on the cell surface. These are called glycan distributions. And we'll ask for how does the complexity of the desired glycan distribution reflect on the complexity of the synthesis machinery. Okay. So to start with, let's talk about the first problem. And what we're going to ask is, how does the precision in the decoding of the noisy morphogen gradient, how does that put constraints on the information channel that you see schematized here? Okay, so at the outset, let me just say that the idea is to sort of extend the scope of information processing and information theory applied to biology in a way that it makes new predictions and is useful in biology. And so, unlike the sort of traditional information theoretic questions asked by Shannon, and in fact, indeed asked by Thomas in the last talk, which is on the maximal efficiency of representing or transmitting information, what we're going to ask is questions about this channel. Okay, we're going to treat the channel as being an active information channel, and we'll ask what are the optimal properties of the channel that ensure the fidelity of the result that you're after. Okay, that drives the fidelity of a global or local objective biological function. So this would be what we're going to do in the encoding part. In the decoding problem, this involves the active information channel that is shown here, okay, which involves the secretory pathway rather than the endocytic pathway that I was pointing out here. And again, one can ask, in order to generate a complex distribution of glycans, what are the constraints on this channel? So the idea would be that we're going to ask questions about, we're going to ask what are the properties of a channel, which is something that Shannon didn't ask, because in Shannon's case, the channel was just a noisy channel, which corrupted the information. Here, the channel is something significant, is functional, and it drives function, and is constrained by the laws of physics and chemistry. Okay? And so that's the kind of questions I'm going to ask. Okay. So to start with the first one, as you heard in the last talk, a major challenge faced during development is how do you reproducibly generate the characteristic anatomy of the organism, starting from the initial form of the embryo. And this has to do with the ultimate ultimate drive to ultimate self-fate decisions, which are made by reading a noisy morphogen profile secreted by producing cells and delivered to the rest of the tissue. Typical equations that go to describe the profile involve diffusion and degradation, giving rise to an exponential decay, as shown here. And since a lot of these, both the diffusion and degradation and the production are noisy, the eventual profile of the morphogen that is received by the rest of the tissue is also going to be noisy. Okay? And the idea is how do you read that noisy information and get such precise developmental outcomes? So this, I'm going to now skip this because this is exactly what Thomas talked about. In fact, this is his work, which shows that the precision of positional inference is accurate up to 1 cell width, or 1% of the cell of 1 cell width, or 1% of the total embryo's length. And it's this hard task that has to be achieved by the tissue. How to convert a very noisy morphogen profile, as which I'll show you in the next slide, a very noisy morphogen profile, and convert it to a much less noisy or very precise developmental outcome. Okay, so now the way we're going to approach this problem is to combine an information theoretic point of view with systems biology, okay, and eventually, hopefully, include mechanics. So the way we pose the problem is to say that the relative positions of the oh, maybe I can show the pointer here. Yeah. Okay. Yeah. I'll just get a pointer there. Can you see that? Yeah. Okay. That the relative position of this of cells in the tissue is encoded in this noisy morphogen profile, and both the production and the transport from this producing cells gives rise to a noisy morphogen profile, which is encoded in this probability distribution P of L given X. Okay? L being the ligand or morphogen. Now, receiving cells then read and process this input to generate a meaningful signaling output, that is P of theta given X here, that's shown here. This involves the generation of this output involves a computation which in turn involves the setting up of a chemical reaction network which reads and processes this information. Okay? The chemical reaction network that every cell is endowed with also has its own noise, and so contributes to an additional intrinsic noise over and above the extrinsic noise that the input distribution of the morphogen had. Okay. Following this, following the computation of the output theta, the inference of the cellular position is obtained by just the application of Bayes' theorem. Therefore, it will give you P of X star given theta, and a maximum a priori estimate to give you what is the inferred position X star. Okay, so with this, one can sort of measure the local inference error, as I show here, as a root mean square deviation from the actual position, and a global inference error here, which is defined as an integration, a weighted integration over the entire tissue. Okay. This minimization is done over, so the idea would be now to minimize this error, and the accuracy and robustness are then defined as being as following. So the idea would be that you would like to see what is the channel, or what is the parameters defining the chemical reaction network that goes towards minimizing this global error, and that the global error, that the parameters so obtained should be robust. That is, if I make slight variations of the parameters, that should not change the inference error at all, or very slightly. Okay? So this globe is a global objective function, whereas the computation is done locally. So that's what I meant by a distributed system. You're locally reading the information from the morphogen, doing the computation, and then and each of these outputs, each of these cellular outputs goes to compute a global inference error which the tissue is trying to minimize. Okay, so now each cell, as I said here, is endowed with multiple receptor types. So this is the phenomenology that has been inspired by a particular signaling system called wingless, which operates in the wing imaginal disc of Drosophila. And what we're going to allow for the existence of multiple surface receptors. We'll call them signaling and non-signaling receptors. This nomenclature will be clear later on. And a chemical reaction network. Okay, so every cell has some level of signaling receptors and non-signaling receptors, and each cell is endowed with a chemical reaction network, as shown here. These chemical reaction networks come with multiple topologies, and therefore parameters that describes the chemical reaction network. So the number of internal processing levels in each cell is going to be described as tiers. The number of receptor types is going to be described as branches. So every cell is endowed with a chemical reaction network with n number of tiers and k number of branches. Okay. Here on the right, I'm showing you the wingless signaling mechanism, which involves the internalization of the morphogen wingless via two different kinds of receptors, frizzled and dally. So they constitute the multiple signaling and non-signaling receptors that I talked about. And it has a certain internal, these two ligand-bound receptors are then internalized within the cell and processed, finally giving rise to a transcription readout. Okay. Now, we check for the performance of each channel in terms of this minimum average inference error. Okay, as I mentioned earlier, and for this we involve we follow this following we follow the optimization strategy that I'm going to tell you here. Each cell is, as I said, is endowed with a certain number of surface receptors, surface receptor types, some amount of signaling receptors and some amount of non-signaling receptors. Okay, so these are called branches. Then each cell is endowed with a chemical reaction network, which has a number of compartments or internalization levels and number of branches that I declared earlier. The chemical reaction network has a specific topology with feedback controls that I represent by the red arrow. And each chemical reaction network has certain rates with the feedback parameters given by Hill coefficients. Finally, using this model for the internal reading of and processing of within each cell, one computes the output P of theta given x, from which one can do the inference and hence the minimization. Okay. Now, so the minimization is going to be run over all the parameters describing the chemical reaction network. Yeah, I just wanted to say that this is typically a very high-dimensional optimization, and it's not the usual working ground for physicists. But if you do have fantastic students and fearless students like Krishnan and Alkesh, who did the work that I'm going to talk about, there's much to be gained by pushing through this unknown territory. And so it's appropriate in this system, in this context, to think of the cellular channel, which is described by the chemical reaction network, as a dynamical system, okay? Because you're giving ordinary differential equations describing the levels of each chemical entity within that within the cell. Okay? And so these dynamical systems are typically stochastic because the input is stochastic, and there is intrinsic noise in this chemical reaction network. Now, the surface receptors that I talked about can be either treated as an open loop control or a closed loop control, and in the second case, you find that the output that is generated can be fed back on the receptor levels, and allowing for a closed loop control. Okay, so you're bringing in the surface receptors as part of the dynamical system. Now, what this gives you, the two things I'd like to mention here is that there is a notion of temporal integration. That is, you're computing the output over a given time interval, and even though I've talked about cell autonomous computation, the fact that the output here can be fed back onto the surface levels gives rise to a sort of cell-cell communication. Okay, and you'll see the effect of that in the eventual way the surface receptors. The profile of the surface receptors is generated. Okay, so let's look at some of the results of this mathematical scheme. We'll first take extrinsic noise alone and ignore the intrinsic noise coming from the chemical reaction network, and we'll consider this very simple and canonical way of reading the information, namely having one cell type, one receptor type, which reads which binds to the ligand, and and degrades, and we allow for a certain feedback. Okay, so this is the plot of the computed inference error as a function of position away from the source, the morphogen source. In black here is the unprocessed inference error. That is, if you don't have any processing, you're just reading the noisy morphogen gradient. And in blue is the reduced inference error obtained by having this kind of architecture. Okay, you can see that this kind of architecture reduces it, but it nowhere comes close to the dashed line, which is what is the desired 1% accuracy. Okay, in this particular architecture, there being only one cell surface receptor type, it simply tracks the morphogen. That is, it decreases away from the source. Okay, now by deploying two different receptor types, something quite dramatic happens. So we'll have now two different receptor types, a signaling receptor and non-signaling receptor, together with the interbranch feedbacks that I showed in the red arrows. And what we find here is that, for instance, this is the open loop control, and this is a closed loop control of the same kind of architecture. What we find here is that there is now a tremendous reduction in the inference, going almost close to the black dashed line, which is the desired 1% accuracy. Now, the surprise here is that is on the receptor profiles and how it looks as a function of position of distance away from the source. Now, recall the previous one where the receptor simply tracked the morphogen. When you have two receptor types, okay, it's the non-signaling receptor that tracks the morphogen, so it decreases away from the source. But the signaling receptor increases away from the source. Okay, this is a surprising result, and it's this particular profile that provides an optimum inference for this network topology. Okay. Now the other important thing was that so this in fact leads to a prediction which can be experimentally tested. Okay. I'll very quickly I'll skip the intrinsic noise for lack of time, but I'll just simply now ask, how does this compare with experiments? And these experiments are done by Chaitra, who was a student in Jitu Mayer's group. And she was looking at the wingless morphogen gradient and how it's received by the cells in the wing imaginal disk. And the relevant, so this is the relevant endocytic pathway. There are two, for wingless, there are two receptors, as I said, a frizzled and DALI, and they enter into the cell via two different endocytic pathways and come together in an early endosome. Now, so this, you can see that this cellular signaling architecture is very similar to the two-branch, two-tier model. Okay. Now, what Krishnan did was to take this model that I showed you earlier, the two-branch, two-tier architecture, and look at the levels of the surface receptors. And I said earlier, what he showed was a signaling receptor increases away from the source, non-signaling receptor decreases away from the source. And this is precisely what Chaitra saw in her experiments. Frizzled, the signaling receptor increased away from the source. DALI, the non-signaling receptor decreased away from the source. Further, what Krishnan could do is that numerically, he could get rid of this particular branch, and the prediction would be that the wild type or would be in blue, very low inference error. The moment you get rid of this branch, the inference error increases. Here, we are taking the coefficient of variation of the output as proxy for the inference error. Okay. Chaitra can do the same. She can get rid of this branch by using a dominant negative version of a key protein, which is involved in the internalization of the non-signaling receptor. And by doing so, she finds that the wild type in blue gets converted to the orange in the mutant. Okay? Very similar to what is predicted by the theory. Okay, do I have 10 minutes at least?
8 minutes.
Okay, so I'll now so far I've talked about the decoding of the complex of a noisy morphogen profile. I'll now switch to the problem of encoding a complex information in the glycans. And this is the scenario that I'm looking at. Proteins which are synthesized in the ER get transferred to the cisternae, the multiple cisternae of a Golgi, whereupon it's sequentially chemically modified using enzymatic reactions. And these are sugar groups that are added onto the proteins. These are called glycans. These glycans are now displayed at the cell surface. Now, so it turns out that the glycans that are synthesized in the Golgi and displayed on the cell surface forms a glycan code. That is, it's a marker of cell type identity and niche identity. And so these glycan codes can be pretty complex, and they're established by looking at mass spec data. And so now, by taking experimental mass spec data and looking at what is the desired output of the glycans that you want to see, one can compare the synthesized glycan distribution to the target desired complex glycan distribution. So again, it's the same thing. The cisternae that I've shown here are endowed with chemical reaction networks. And you ask for, which have a certain set of parameters, topologies, feedback control, etc. You optimize all this to see how close is this synthesized glycan distribution to the target distribution. Okay, and so you get an optimal network and you can ask questions like, if I increase the complexity of the target distribution, how much more complex should the synthesis machinery be in order for the synthesized glycan distribution to be as close to the target distribution. Okay, and this can be done using. Sorry. Okay. That's not here. Okay. Okay. So with that intermission, so in both the decoding and encoding problems, the optimization that we noticed is a very high dimensional one. There are lots of parameters, different topologies, feedback parameters, etc., etc. And so it's a very high dimensional optimization, which is in general highly non-convex, and therefore supports multiple minima. So what we'd like to do now is that once you hit upon the optimum, you can say how does the inference change, or when I change the parameters ever so slightly. So to do this, we studied the geometry of this inference landscape, and I'll take the information decoding problem as an example. Okay, so the way we do it is to look at go to the global minimum and construct the Hessian or the curvature of that global minima. Okay, being a high-dimensional optimization with many parameters, the Hessian is described by a matrix which is high-dimensional, and what we find here is that, to cut a long story short, is that most of the eigenvalues of this Hessian are very low. That is, if I move along these different directions, I don't encounter any curvature. Okay, so they are flat directions, and a few of them are stiff. So, for instance, an example here, the red dot here. So this is a plot of the signaling versus non-signaling receptor levels on the cell surface, and the red dot tells you the optimum one on a particular cell. Okay. Now, if I make variations along the non-signaling receptor in this arrow, then you see that the inference error doesn't change much. However, if I make it orthogonally along the signaling direction, it changes a lot. Okay? So the properties of the signaling receptor are very stiff, rigid. They have to be tightly controlled in order to keep the inference robust. Whereas the non-signaling receptor is allowed to be sloppy and free. Okay? Any variation along that, the system does not care because the inference error doesn't change too much. Similarly, if I look at the binding rate of the signaling receptor, it's very sharply peaked at one end of the allowed distribution of ligand binding rates, whereas the non-signaling receptor is much more promiscuous. Hence, justifying the name that this particular receptor is actually non-signaling, is very promiscuous, can bind to many different kinds of ligands, whereas the signaling receptor is much more constrained or fixed. Okay, so in other words, the signaling receptor is specific and is along variations of the properties of signaling receptor reflect a stiff direction. The non-signaling receptor is non-specific, is promiscuous. It can bind to many different morphogens, and variations of its characteristics is very sloppy. Okay. A similar analysis can be done to a similar analysis can be done by looking at all the, remember it's a non-convex optimization, so there are lots of minima, and what you can do is first go to the global minima. You find that there are most eigenvalues of the Hessian are low, shown here, okay, and very few of them are stiff. Okay, which means that the global minima has many shallow flat directions and very few high curvature directions. Okay, since it's a non-convex optimization, there are many different minima, and you can ask where do these other minima lie, and surprisingly, what we find is that the other minima lie on the sloppy manifold. Okay, so if you look at the position vector from the sort of position vector of the other minima relative to the global minima, then they all are co-aligned with the sloppy directions. Okay? So not only are most directions of the global minima sloppy, but all the other minima lie on the sloppy manifold. Okay. So this gives rise to an interesting general problem which we might try to explore. And it is the following, that in both these examples and many other examples that we have been looking at, in general, these examples are all described by very complex, high-dimensional chemical reaction networks, for which one may define an objective functional, which is a functional of the parameters, the topology, and the noisy input. The scope of this calculation was to show that one needs to minimize this objective function in the space of parameters and topologies. Okay? And that's what I'll call F star. This optimization is very high dimensional, as I said, and is non-convex. There are multiple minima. And I described the way we entertained such a result. Okay? So ultimately, the idea is that the topography of the low energy, of the low inference error landscape is something like this. Okay? Most directions are very sloppy. You're moving around in the valley, but there are few directions which are stiff. Okay? And these few stiff directions need to be subject to cellular control. Okay? Now, the question is, how does such high dimensional, or what is the reason for having such a high dimensional reduction? How common is such a dimensional reduction in a variety of biological networks that we might see? And yeah. So I'll just end very quickly. And what I'd like to maybe I'll just very quickly say that if I look at in the decoding problem, if I plot the signaling receptor versus the non-signaling receptor here, then what you find is that if I look at this space and ask what is the value of the signaling receptor and non-signaling receptor that optimizes a local inference, then it's given by that point here, shown in red there. Okay, but if I ask now, what is the where in this space does the global optimization lie? It's in the green line. Okay, so the local optimization is very different from the global optimization. That's the point. And each of these optimizations has their own sloppy manifolds. And the idea that I'm trying to investigate now is the notion of evolvability. The reason why you have such high dimensional sloppy manifolds is that it allows for an intersection of the sort of sloppy manifolds of these two objectives, the local objective and the global objective. Okay? And if indeed it was a low dimensional, then the probability of such intersections would be very low. Okay? So the idea is that the reason for dimensional reduction in these parameters is because of a desired biological feature, namely evolvability. Okay. So with that, I'm going to end my talk. And Krishnan here did all the beautiful work on the information decoding problem, ably assisted by Chaitra, who is a student in Jeetu Mayer's lab. And these are the references. The problem that I just breezed through on the Golgi and how the properties of the Golgi synthesis machinery are constrained by the complexity of the output that you want to generate was done by Alkesh, who is another student, in collaboration with Quentin, who is a student of Pierre here, and my long-term collaborator and information theorist, Garud Iyengar from Columbia University. And with that, I'll thank you all.